What is a stem cell? It is a cell that can mature into different types of adult cells. This property is known as pluripotency, and the cell is said to be pluripotent. A stem cell is like a young child who has many career options open to them. As they grow older, their career paths become more and more restrictive, and eventually they focus on one particular area. A stem cell also goes through a similar phase as it decides what type of cell to become. While people cannot revert back to children, an adult cell can become an induced pluripotent stem cell or iPSC and then change into a different type of adult cell. All genetic information of a cell is stored in the DNA, which is like a library. A gene is like a book that contains instructions for the production of one type of protein. Proteins are responsible for maintaining the characteristics of cells and also determine the fate of stem cells. Histones are proteins that hold DNA together. They can be pictured as the outer walls of the DNA library. As a stem cell matures, it undergoes epigenetic modification. Methyl tags are added to regions of DNA which will no longer be read and to histones to regulate accessibility of genes. Also, acetyl tags are put in particular regions of histones to increase likelihood of genes being read. When an adult cell is reprogrammed into an iPSC, these epigenetic changes are reset. Methyl tags from DNA are removed and changes are made to histones, so genes associated with pluripotency are now read. This was first achieved by artificially introducing a set of genes known as transgenes coding for the proteins OCT4, SOX2, KLF4 and CMYC using viruses as vectors. The first stage of reprogramming is called initiation, and at this stage the cell is dependent on the transgenes. OCT4, SOX2 and KLF4 can move through closed regions of DNA and increase the probability of pluripotency associated genes being read. CMYC can help them bind to certain parts of DNA. The next stage, maturation, begins when the first pluripotency associated gene becomes active and gradually more follow. At this point, the cell starts producing its own pluripotency related proteins like OCT4 itself, which helps to reprogram the cell. This means it starts gaining stem cell characteristics and starts becoming independent from the transgenes. The final stage is known as stabilization, and this is when another set of pluripotency related genes become active. OSKM are no longer required for the cell to remain pluripotent. Epigenetic changes occur that result in iPSC resembling stem cells. iPSC are a breakthrough in biology as they can potentially be used in regenerative medicine like synthesizing human organs in a laboratory for organ transplant and even treating Parkinson's disease. They are also suitable for research purposes because of the ethical concerns surrounding embryonic stem cells. Thank you.